So hello everyone, really nice to see you all and for coming today to this PCP midweek mixer. So I think most people know me. So I'm Lamia Losin, research scientist at Agriculture and Agri-Food Canada in Saint Hyacinthe, and currently the PCP division chair. So first, before to start, I would like to thank also AOCS to enable this event, and Dr. Lee Yun Chen, who is the PCP co-chair from the University of Alberta, and Kostav Majumdar from the University of Nebraska, really to helping in the organization of this event. So just before to start, I would like to communicate some housekeeping items. So definitely we will really appreciate if you could turn on your camera, mute your microphone, and put your name in the add your affiliation just to know you and get connected with others. And of course, introduce yourself. And if you have any question during the presentation, you can always raise your hand at the end or put typing it into the chat. So, yeah. So today mixer actually is about the utilization of advanced emerging processing technology. We thought this is a very...
You are muted, Lamia. Hi, Lamia, you are muted.
Did you hear me? I was muted all the time?
No, just
no.
Okay, just I don't know what happened. It decided to mute me. I don't know what happened. I didn't push anything. But anyway, so as I was saying, it's today mixer is about advanced processing. We thought that this is a very hot topic to touch on today. And because these technologies have been identified as versatile and clean and eco-efficient processes that not only can preserve food, but can also improve their nutritional quality as well as their functionality. So we are really lucky and thrilled to have today two experts that are going to touch on this topic and give us their thought. It's the Professor Vijaya Raghavan from University of McGill University, and Professor Pam Ismail from the University of Minnesota. So the speakers will talk about 15 minutes, and we will have about 10 minutes for the question, for the Q&A. And so we hope the things will go as planned. So first, I would like to introduce Professor Raghavan, who is a James McGill professor in the Department of Bioresource Engineering from McGill University, Quebec. He was inducted into the Royal Society of Canada in 2012 and into the Engineering Institute of Canada in 2017. He was also the president of the Canadian Society for Bio.
You are muted again, Lamia.
You got muted.
Who is muting me? That's very strange. Nothing, I don't know. But anyway.
Somebody doesn't like you, looks like. I'm sorry.
Looks like someone.
I'm just kidding.
I don't know what's happening here. But anyways, so I don't know where did you stop hearing me, but I was just saying that Dr. Raghavan is the president of the Academy of Science of the Royal Society of Canada from 2017 to 2019. He has made a huge contribution to the development of knowledge and technologies in the area of post-production technologies related to drying, storage, thermal and non-thermal treatments of food, as well as the valorization of agricultural and food bioproduct, leading to more than 630 articles, 53 book chapters, and 7 patents. He is studying the use of electro technologies such as microwave, radio frequency, pulsed electric fields, not only for the drying and the thermal treatment, but also for the extraction of bioactive compounds from plant material. So please, Dr. Raghavan, the screen is yours.
Thank you very much. Can you hear me okay? Yes, certainly. Thank you for the kind words of for the audience, it's all what I'm going to do in the next few minutes. That's the most important part, isn't it? So nice to see you, and thank you for inviting me to share some of my thoughts with you. So certainly the time that's available for me is pretty short, but certainly emerging technologies could be pretty huge. So I'd like to share what we have been dabbling with over the years, and also some of the philosophies behind that. So with that, I would like to share my screen with you, the PowerPoints. So I go directly to that. So can you see the slides okay?
Yeah.
Okay. Well, first of all, it's appropriate for me to acknowledge the land that we are in. So McGill University, it is on a land which has long served as a site of meeting and exchange amongst indigenous peoples, including the Haudenosaunee and Anishinaabe nations. We acknowledge and thank the diverse indigenous peoples whose presence works this territory on which peoples of the world now gather. So with that, I can change my slide. I wonder why. Oh no, yes, now I can share. Okay.
Okay.
It's a bit of a lag
Yeah.
involved. So I just like to share, McGill University is in the heart of the downtown Montreal. I'm in the Faculty of Agriculture and Environmental Sciences, which is the tip of the island, and that's what you are seeing here. So the topic of interest today is the emerging technologies, and certainly there are many things that has happened with us, and then it's all connected with respect to the food systems. So when we say food system, the current challenges that we have is with respect to the population increase and also the availability. So food security weighs in. And when we are doing the food systems, so there is a significant amount of loss that happens. So it is very important for us to take care of that losses as well in an efficient way. Therefore, technology is important for that aspect as well. So our research interest, definitely, it encompasses the whole food system. So therefore, certainly post-production all the way to the utilization of that. So to address those issues, we have to be environment friendly, and also food safety is important to produce high quality products, which is safe for the consumers. So the technology is what we are focusing on. So technology, I would like to classify into electro technology. So electro technology is becoming more and more interesting, especially when you talk about the efficiency point of view. So if I take drying, for instance, so can we, drying involves the phase change. Phase change from the thermodynamic point of view is very energy consumptive. So therefore, pundits are looking into possibilities of integrating the electro technology to that. So it is the same for food processing as well. So if you look at the electrical aspect, as well as the other aspect like the ultrasound, pulse light, and ultralight. So they're all useful in many ways, especially when the society is looking for the plant-based food. So when you want to have a plant-based food, so we always need to integrate emerging technologies in whichever way we can to make it much more efficient and user-friendly. So in my lab, I just want to share with you, we do work quite a bit on the microwaves. So for us, so we normally design equipment suitable for the research aspects that we need to have. And that's what you are seeing here in this. The red box is actually the microwave where I can change the frequencies for that. And then I can connect that up into the single mode or the multi-mode cavity where I can keep track of the incident power and the reflected power. Thereby, I can actually calculate the absorbed power. So in a similar fashion, we also do radio frequency and also the pulse electric field. And in these kind of systems, I can actually integrate. If I want to do the convective drying, for instance, I can do that along with the microwave. So those are the flexibilities that we have created ourselves in the lab, and to be able to understand more and more of the complex food matrix that we are going to be confronted with. So with respect to the thermal treatment, certainly microwave, to understand the dielectric properties, how it can be utilized, we have dealt with many, many products, not only for specific food processing, but also extracting actually the molecules. The molecules that I can get from the food material is important, and for that, certainly microwave extraction helps for that. One of the important work that we have done is the in-shell pasteurization of eggs. So in-shell pasteurization of eggs, if we want to do it in a conventional way, it is difficult to achieve the egg white, which requires a lower temperature as opposed to that of the egg yolk, if you want to talk about the pasteurization part of it. So we immediately went into the microwave system, where we could put our numerical methods as well as the experimental validation to come up with the right kind of the waveguide, so that I can achieve the requirement of the pasteurization for the in-shell egg. So that is something which is still has further development that could be done. And one of my students is working in US. And so we are collaborating to see we can take this, especially the microwave technology is also changing very fast, and we collaborate in that framework. So in a similar fashion, you can also do the combined microwave hot air or vacuum drying aspect to maintain the good product quality, and also especially when you are trying to use the sensitive products in the food systems. So in a similar fashion, we do focus on the pulse electric field. So in the pulse electric field, one of the projects that we did on pasteurization of the liquid eggs. So for doing the liquid egg, so we did have the pulse electric field, but then the treatment chamber is what you are seeing here, where we can continually circulate the liquid egg so that we can achieve the necessary energy to get rid of the microbe, which is what is involved in the pasteurization of it in there. So as we all know, depending upon the magnitude of the voltage that you have, you can either create induction stress, or you can improve mass transfer, or you can do microbial inactivation. So we have over the years learned to design these things, and then have the equipment available for us to do different attributes or the different commodities that we are going to be studying with that. So electrohydrodynamics, especially now, there are many groups, research groups are looking into it, because the electric wind, though, especially when you start talking about the physics of the electrohydrodynamics, it's very interesting. And then we have experimentally, we have seen that it is possible to get rid of water in a very efficient fashion. And the thermodynamics of it is still, I'm trying to understand that in a global fashion in that. So in this electrohydrodynamics, which is useful not only for the drying, but also to maintain the good quality material when you are done with the entire process. So those are some of the things that we are looking at. And when you are looking at the waste, so certainly the waste, pyrolysis becomes one of the important avenue for us to do that. So for pyrolysis, I can generate biochar, which can help us identify the carbon sequestration possible in that pathway. So it is that is another area where we are trying to look at the waste management aspect of it, biochar as well as hydrochar. So they are two things which are very useful for handling waste products. So I just want to spend a little bit more time on the particulate medium processor. This fits into the category of enhanced heat transfer. So basically, we all know conduction heat transfer, but the particulate medium, I can have, say, for example, salt. I can submerge some agricultural material within that. So therefore, because of the product characteristics, I can exchange heat in a much faster fashion. So using this particulate medium processing techniques, you can do either heating or cooling or freezing, so on and so forth. So these are all possible. So therefore, the application point of view, it can be thermal processing applications, or you can do the disinfestation, degermination, popping and roasting, so on and so forth. So in that, as soon as you understand the particulate medium heat transfer, certainly the equipment design becomes an important area. So over the years, I have been involved in coming up with, here is a sectional view of where I can feed in on the right-hand side, the agricultural products which comes into contact with the particulate medium. In the midsection, they can be mixed together and they can be separated, and then the job is done. So that is all right, but then how do I scale it up? So we always have that problem. So therefore, we looked at independently doing that. So here, what you are seeing on the left-hand side, I can heat the medium in a separate place, and then I can bring them together with the particulates, and I can separate them, and I can recycle the medium. So therefore, it becomes much more efficient. So we have done the convective. On the right-hand side, you are seeing the cross-flow convective dryer. So therefore, we have always compared with the convective versus the particulate medium to get some of the attributes, which of course is easily scalable. Here is the equipment which can do 1 to 3 ton per hour capacity in that. So some of the advantages that we have seen in the grain preheating, it can be 5 times faster and 87% more efficient. And in roasting, certainly some of the equipment that is available in the industry, the system, the roasting soybeans can be done at 500 kilograms per hour. And of course, it's scalable to any amount that you would like. Thermal disinfestation can be done, degermination can be done. Of course, if I want to do the drying, then I have another option to bring that medium. I can have the selectivity of the medium for doing this. That is also another advantage of this. So now the protein for the plant-based food is most important. I think particulate medium processor can help us. And here, let's say, for example, P, they have some negative aspect attributes to that. So we can get rid of that negative attributes by using the particulate medium processors so that you can denature some of the enzymes which are related to the off flavors, and then you can have a good protein source for that. So I'm trying to see whether we can couple that, especially with the demand for protein that is available, that is necessary for us, especially when you're looking at the plant-based food. So that's something which we have been doing. And there are a number of references here. There are many publications that we have come through with all of our research work to share with our colleagues in there. With that, I would like to take a moment to acknowledge all the funding agencies that have gone through, and also the graduate students who have worked over the years in these interesting, challenging problems to learn more of that, more of the mechanisms involved. And certainly with that, I would like to thank you all for listening. And I'd like to share this. The biggest innovation, of course, is connected with the biology and technology. And that's where we are here at this point. With that, I am ready to answer any questions if I can. Thank you.
Thank you, Dr. Raghavan. Very interesting to show first the extent of your experience, but also what can be achieved with some of these innovative and advanced technologies. Please, if you have any question, raise your hand, or you can put the question in the chat. Actually, while waiting for some participant questions, I would like to ask you, what is the readiness actually of these advanced technologies in terms of industrial uptake of these technologies? Do you think we are at the point where it is easily applicable these days, or it is still at the research state?
Well, as we all know, certainly the microwave technology is very much used industrially, but then the question is whether, what kind of a frequency you want to be looking at. So most of the research data that you see, scientifically speaking, they are all related to our kitchen microwave frequency, 2450 megahertz. So like 40 megahertz, industrially speaking, you can have a very high level, so high throughput possible with the higher frequency range from the microwave perspective. So therefore, that frequency, the research data that are available is pretty much on the 2450 at the kitchen microwave. So we need more of that kind of data, especially. But then industrially, it is already being used, and scaling up is one of the important criteria that we always have to be focusing on. So as I mentioned to you, if you want to do drying, for example, drying is always very energy consumptive process. So especially in the agricultural production systems, if you want to look at it, most of the drying is based on the convective system, for example, corn drying. So now, how can we bring the electro technology to that? That is one of the challenges as far as the futuristic point of view. And I know there are several groups that are looking at the possibilities of doing that. But then from the food nutritional point of view, especially if there is a food material which is not ready to be sold at the consumer level, I can always attempt to extract active ingredients from them. So that's one aspect which has to happen. And also the bioavailability of the nutrients to the humans. So therefore, there are gaps at that point, and then slowly the research efforts are being put in to understand those mechanisms, especially in today's context. It is all multidisciplinary in nature. So therefore, how do we actually feel comfortable to be connected with the other areas that we are not comfortable with, and how to understand that, how to integrate them, and how do we make that possible? And then there are a lot of interesting challenges that we have at this point.
So interesting to see that what you are proposing is that the advanced technology could have an important role to play in improving, for example, the utilization of some co-products, on adding the value to those co-products, which conventional technologies won't enable, right? That's your thought.
Yeah.
I have another question from Dr. Professor Buddhi Lamsal from Iowa University. He is asking about the impact of such novel technology on the structure and function of plant proteins, that he thinks it will be important to evaluate. So he would like if you could share your thoughts on this aspect.
Yeah, certainly the plant protein, I think one of the major efforts that we are trying to do is we have seen, if I want to do the protein modeling, so certainly I can take the crystalline structure of the protein and apply the necessary stresses onto it. And then you can see the unfolding of it in an interesting way. I can do those modeling aspects for a very short period of time. But then in the lab, when I do it, I can actually have the actual real-time approach. So when I do the real-time approach, we have seen that I can, by altering the protein structure, I can take away the allergenicity attributes from it. Mm-hmm. So that is one aspect of it. But also we can make the food more bioavailable. So there are parameters associated with the bioavailability aspect of it. And that is also we are examining, especially from the modeling side. And when I do the modeling, can I actually come up with nanoseconds into the more realistic times? So then, of course, you need a lot of computing power, and that's what we are trying to work on, to understand that modeling approach and then actually do the same thing with respect to the experiment in the lab.
Yeah, so interesting.
So now, in today's context, we are looking at how do I actually have a plant-based food. When I look at the plant-based food, it is always that protein part which has to be, which falls short of. So how do we actually get that? So from our Canadian perspective, we do have lots of pulses that are grown, which are rich in protein. So therefore, how do we actually make that protein? It is a big industry. It is possible to do, and that will be very, very important, especially for the plant-based newer products that are going to come out.
Interesting to see the impact of these technologies on the allergenicity of food. Particularly, this is becoming I mean, it's an issue. Introducing new proteins always brings also some allergenic risk with that. We have a question, I think, from, actually you have many questions. I don't know the time, but Leon, you had maybe raised your hand some time ago.
Yeah, thanks to Dr. Raghavan for this eye-opening presentation. I'm especially interested in the particulate medium processing, and you specifically mentioned that it can be used to treat bean to remove the beany flavor. Could you further comment on that? How efficient they can remove beany flavor? What's the mechanism? Also, I'm interested to know the particle as the medium. What are the particles you use for this processing?
Well, you know, this is an old technology in some ways. It is no way emerging technology, because way back when we were trying to deal with soybean to be fed to the dairy cows, you cannot feed in the raw soybean. It has to be thermally treated. So at that point, when we started working on this particulate medium method, the competing technology in the industry, they were putting the soybean through flame. So it was denaturing the trypsin inhibitor, and also it was denaturing some of the other good proteins. So our challenge was, can we come up with something, the particulate medium, say for example, sand. So sand is a particulate medium. So I can actually do this when I have a certain temperature of the sand. I can actually bring soybean and mix them together and separate them out. So sand, from the heat capacity point of view, it has a higher number with respect to the air. So air is easy to get, but it has a poor heat capacity. So therefore, from the heat transfer perspective, you can have an enhanced heat transfer situation for particulate medium. So that was the driving force for us. So then you can do many different things. You can have some thermal condition for the bed, and you can have the delta T that is available from the whatever agricultural material you want to process. You can configure that in any which way you want to. You can roast. You can do popcorn if you like. So for example, that machine that I have showed you, I can do popcorn pretty much with that high capacity, because I can control many things in that. So that is basically what we are looking at. So conduction, convection, radiation processes, here I can do the particulate medium. It kind of fits into the conduction mode, but the effective heat transfer is going to be much higher. And then, now I'm trying to involve with the team, you know, the protein group in Canada, for example, see whether we can take this, because we are all looking at the high protein content that are necessary. So by dry heat, I can actually process them. And so therefore, other way, okay, if you start with soybean, certainly I can get about 30% of whatever material that I have as protein, right? So therefore, that can be easily adaptable to whatever other users that you want to see.
Very interesting, really. Very nice discussion. Actually, we have many questions in the chat. Unfortunately, because of time limitation, we cannot go through all of them. But please, Dr. Raghavan, you can look at them and feel if you could answer directly on the chat.
Okay.
Well, we will be moving. If we have some time at the end of the presentations of Dr. Ismail, we might go back. But I would like to give now the floor to Leon to present Dr. Pam Ismail and move on with the second presentation. Thank you very much. Thank you for a very nice discussion.
Welcome.
Okay. My name is Ling Chen. I'm the Vice Chair of PCP Division. It's my great pleasure to introduce our second speaker, Dr. Pam Ismail. Many of you know Dr. Ismail already from their, Planet Protein Innovation Centers through their Spotlight annual meetings or newsletters. Dr. Ismail is the founder and the director of this center, and she's also a professor in the Department of Food Science and Nutrition at the University of Minnesota. Dr. Ismail has over 25 years of experience in food chemistry research, including analytical chemistry, protein chemistry, and chemistry and the fate of bioactive food component. Her recent research focused more on the structural characterizations and enhancement of functionality of plant proteins following their new technology processing, and also her research emphasized on protein safety, bioavailability, and also other bioactivities. Due to her excellent research contributions, Dr. Ismail is the recipient of a Distinguished Teaching Award and also an Outstanding Professor Award. Let's welcome Dr. Ismail for her presentation.
Thank you, Lingyun, for the wonderful introduction and the opportunity to speak today. It's my pleasure to talk to you a little bit and hone in onto the plant protein side of things. And due to time constraints, I will just highlight few of the research and the technologies we're working on at the Plant Protein Innovation Center. So this is just a snapshot of what's going on. So I will start here by just briefly, why are plant protein challenging? So why are they gaining that much attention? And people are facing opportunities as well as challenges working with them. So if we think about plant proteins, so they differ from animal-based proteins in so many regards. They're the way they associate, the way they form their structure, tertiary and quaternary structure, and also their love-hate relationship with water. So plant proteins often suffer from not being water soluble, and that impacts a lot of the functional properties. So also processing, we just heard a few examples of innovative processing technologies, some of which might impact the protein denaturation state. And when the protein denatures, it basically exposes more of their hydrophobic characteristics. And with that, we might have protein-protein polymerization, aggregation, and loss of functionality. So in general, when we talk about functionality, what are we really looking for? We think about applications. So we're looking for high protein beverage applications. Okay, so we need a highly soluble protein so that we can include them at a relatively high percentage so that we can put a claim on the label. We want them to form gels in certain food systems. So we want them to form these three-dimensional network and hold water and give us this characteristic structure in certain foods. We want them to emulsify. So for example, we want to form these plant-based cream cheese, and it's like high fat and aqueous medium. So we need a good emulsification. We need a good viscous interspace between the droplets. So we're looking for proteins to aid in that. We're also now the high moisture meat alternative is another rising application where we want these proteins to form fibrous structure and also hold water within. So a lot of demands from these proteins. So we need to look at ways to make them function in so many different ways. So I'll give you again, like I said, just few examples with a little bit of results, not a lot, just to highlight what we're doing. So first of all, it's as simple as optimizing how we take out the protein from the complex matrices. So there are common practices in industry, and what we try to do here at the Plant Protein Innovation Center is test processes on bench and then scaling up in the pilot plant to optimize the process in a way where we preserve structural integrity of the protein. So we have here are two examples of extractions. One is dilute salt coupled with membrane filtration and an alkaline base extraction. Both gave us good purity and very good reasonable protein yield. Along with that here, so those of you that are familiar, with gel electrophoresis, we look at polymerization of the protein. So here in this lane, we have pea protein, and here we have soy protein under non-reducing and reducing. Under non-reducing conditions, we see a lot of high polymers and aggregates at the top of the gel. While the proteins we form in the pilot plant under two different methods lack these high molecular weight aggregates. So we have preserved somewhat the protein structure and limited the formation of these high molecular weight aggregates that even after reducing, after breaking down disulfide bonds, that is, we still see them in the gel. And these again are commercial versus what we produce in the pilot plant. So looking at some functionalities, we can see here, we always compare to whey protein isolate because it is the go-to protein for high protein beverages. So it has high solubility across different pHs, the neutral and the acidic, whether heated or non-heated. And if we look at commercial soy and commercial pea, they have poor solubility, especially in acidic environments. However, the samples we produced have really good solubility, especially the salt extracted with almost 90% solubility. So pretty impressive, and that is at 5% protein inclusion. In terms of gelation, we form a gel, thermally induced gel, and we use Dr analyzer to rupture the gel, and we monitor the force that it takes to rupture. The more force needed, the stronger the gel. Again, we see that commercial pea does not form a gel at 15% protein compared to a commercial soy, whereas our salt extracted pea protein isolate have very similar gel strengths to a commercial soy protein. In terms of emulsification, we see improved emulsification compared to a commercial isolate, whereas we're still not that close to soy. And here we measure just the emulsification capacity, which is the amount of oil emulsified per gram of protein. Looking at different technologies, here we're looking at thermal, non-thermal processing by cold plasma. The work here has been focusing on different types of cold plasma and the interface with protein and how the protein structure changes and how functionality is impacted. So cold plasma is used in industry for surface modification and material processing, for ozone generation, for water disinfection. And it's used or investigated as means of wound healing, cancer treatment, and non-thermal processing of foods. It preserves quality, it is low energy and low cost, it's safe, and it's environmentally friendly. So it warrants further investigation. So we do have one publication from this work and another one submitted, but I'll share with you a little bit the findings here. Just what is cold plasma? Plasma is the fourth state of matter, and the plasma, in the plasma, we see different reactive species. We have oxygen reactive species, nitrogen reactive species, electrons, and the composition and type of species that we see in plasma is dependent on the gas used to generate the plasma, and also is dependent on the geometry of the plasma generator and the mode of contact, whether it's direct or indirect contact with the sample. That means above the sample or directly in the sample solution. The common reactions upon cold plasma is mostly oxidation, where if we have sulfide groups, they're going to form disulfide linkages. We see also bond cleavage. We see unfolding of the protein, exposure of reactive groups, and potentially formation of soluble aggregates if controlled, and then insoluble aggregate if not controlled. So briefly, we looked at different geometries and different gases. And one of the most common geometry is two-dimensional dielectric treatment discharge. So 2D DBD. And we have the atmospheric pressure plasma jet, and we have the nanosecond pulsed plasma. So 3 different configurations, and we looked at the impact on a 5% protein solution. We looked at protein denaturation at different treatment times, and we see unfolding of the protein, a decrease in enthalpy with increase in treatment time across all 3 different geometries. With that came the production of soluble aggregates. We confirmed that with gel electrophoresis as well as size exclusion chromatography, but here just to visualize without treatment and with treatment, we see increase in the high molecular weight polymers that still go through the gel, meaning they're mostly soluble aggregates. And when we see similar across different treatment with some potential formation of low molecular weight peptides due to bond cleavage. But just briefly, few functionalities here, just a couple. Gelation, with all of the treatment, we saw a significant increase in gel strength compared to a commercial product. Here we formulated the commercial product at 20% protein in order to form a gel, and native control, as well as all the treatments. And we saw also a significant increase in emulsification capacity. So we looked further into emulsion stability by forming emulsion with 2-stage homogenizer and storage of, under accelerated storage conditions, and looking at particle size, zeta potential, and turbidity, as well as CLSM imaging. So we see here the commercial product over storage time starts forming large particles. So we see that means we have coalescence and formation of large oil droplets. With the control, we saw some of that happening as well. However, with all of our 3 different treatments, we didn't see change in particle size. And also we saw a minimal decrease in the surface charge on the particle, on the droplet surface. And that maintains electrostatic repulsion so that we reduce the chances of coalescence, whereas we saw a major reduction in the zeta potential for commercial and for the control as well. When we did confocal laser scanning microscopy imaging, we also looked at the particles, and the green stain is for protein and the red stain is for oil. And we see that the commercial product form aggregated portions in the emulsion, whereas the treated samples remained very fine particle throughout the 10 days of storage. Another one that I will briefly talk about, we filed a patent here, and this is to improve solubility of pea protein in particular using Maillard-induced glycation, utilizing the starch from the pea flour so that we don't have to add an ingredient to the mix. And during the production of the protein isolate, we wanted to initiate glycation. So it's kind of like during processing, we functionalize the protein. So the Maillard reaction is basically a condensation of the reducing end of the reducing carbohydrate, the carbonyl group, and the amine group. And the first stable compound is the amidori compound, which is the conjugated or glycated protein, and we control the reaction to prevent it from propagating into advanced stages. So the idea here is when we partially glycate, we have this steric hindrance with the bulky carbohydrate group, and also we increase the net negative charge on the protein surface. So we enhance solivation and repulsion between molecules, and we also enhance thermal stability by imparting a higher net negative charge on the surface. We characterize the product that we, once we hydrolyze to produce the carbohydrate of the specific reducing power, we look for a DE value of 10 to 15 to have controlled glycation so that it's not very fast, high rate, so we can control it actually. So we characterize that. We characterize the degree of polymerization and the reducing power, and we select the conditions that gives us the DE of about 10 to 15. And we look at the, after glycating with this hydrolyzed starch, we look at color to make sure that we didn't induce any browning, and we can see there's no significant difference in the lightness. And we also looked to see how much amine groups we blocked, given that lysine is an essential amino acid. So we got a moderate reduction in amine group upon glycation. We confirmed the glycation by glycostaining, and we can see that the bands became a little higher than the regular band. That's because of the association of the carbohydrate. So we see a shift in the molecular weight, and then we confirm glycation by the glycostaining. And when we looked at solubility, we improved solubility greatly. This is our native non-processed pea protein isolate. We almost doubled the solubility after heating at pH 3.4, again of a 5% protein solution. Last one I'm going to mention here is another project we have going on to functionalize pulse proteins for high moisture meat alternative products. And what we do is we look at chickpea and pea protein, and we produce them in the pilot plant under optimized conditions. And then we functionalize them using transglutaminase to produce high molecular weight polymers. And we texturize, and then we do sensory analysis as well as textural analysis on the final product. So here just briefly, transglutaminase, some of you know, is an enzyme that stimulates the covalent linkage between a carboxyamide of a glutamine and an amino group of a lysine to form a covalent linkage. So enhance the formation of large molecular weight polymers that will aid in enhancing gelation. So far you're showing that with transglutaminase, we see an enhancement in gelation, and we are in the process of texturizing these different proteins. Oh, I lied. One last one is breeding. So we're looking to enhance the quality of pea protein through breeding by promoting changes in the legumin to viscellin ratio. So we're studying the impact of changing the ratio of legumin to viscellin by fractionating these proteins and recombining them at different ratios, and then looking what happens to structure and functional properties. And we're isolating these fractions and using them to produce a high throughput FTIR method to predict profile in hundreds of accessions so that we can aid the breeding program. We are assessing nutrition quality as well as functionality, and then initiating finally a breeding program with targeted ratio of legumin to viscellin. So very briefly here, we are fractionating the viscellin from legumin in very excessive and lengthy biochemical processing or fractionation, and we're recombining them at different ratios and characterizing them. But at the same time, we're taking these rich fractions and using two-dimensional liquid chromatography to purify them further and confirm their identity by proteomics so that we can develop the FTIR prediction models. So basically, we aim as to have new quality-driven protein isolation protocols, to have function-enhanced protein isolates, to provide feedback to breeders, as well as economic prosperity to farmers in the food industry. And the last takeaway I have for you is plant protein Demand is steep and long-term, not going anywhere. And while plant proteins are challenging to work with, there's always room for improvement, and the research in this space is ongoing. With that, I would like to thank all my students and researchers, and thank you for listening. And I think we have a few minutes for questions.
Thank you so much, Dr. Ismail, for this excellent presentation. Thanks for sharing with us the new method, new technology that can modulate protein structures and bring new functionalities. I also appreciate your insight about future perspectives and encourage us to continue our journey to develop new plant protein food. I would encourage our audience to ask the questions. While waiting for the questions from the audience, I can start with one question. Dr. Ismail, that's very interesting. You see, after cold plasma treatment, the emulsion droplet become more uniform and they don't have coalescence and fluctuations. You mentioned the charge is one factor because the charge increased. Do you think there's any factor related to the protein unfolding? So how would you see? That's quite interesting. I wanted to see the more mechanisms about this improve the functionality.
Yeah, thank you. We are currently pursuing the more molecular interactions at the interface. So this is still a research ongoing so that we can understand better the mechanism, but definitely there is protein unfolding. We saw that with looking at denaturation by DSC. We saw that due to the formation of soluble aggregates. So there is some protein unfolding. And what happens is at the interface, so when the protein forms this thin film at the interface and orientate the polarity and the charges to the surface, it was apparent that the modified protein retained to some extent their negative charge on the surface because there was less, coalescence forming. So once there are coalescence, then you will detect less charge on the surface because of the formation of larger particles. But the mechanism of that will require really further deeper dive into the molecular.
Thank you, Dr. Ismail. I see Janita has a question. Raise her hand. Janita, you can unmute yourself.
Yeah, thank you very much, Pam, for your excellent insight into these changes that can happen with the technologies. My question is related to the fiber formation. So you are using transglutaminase to cross-link and then basically use high moisture extrusion to make them into a fiber, right?
Well, that is ongoing. So we are forming the protein isolates in the pipe plant, and then we are modifying them. And so far we're doing structural and we enhance the gelation. The next step is texturizing with our benchtop extruder.
Okay, so basically globular proteins, you enhance the cross-linking and it could be string and bead kind of network formation, right? Then through mechanical processing, change it lengthwise or change into a fiber-like nature. Okay, right. Yeah, don't be clear on that. Okay,
yeah,
thanks.
Yeah, that's the intent. The intent is to enhance the potential of fibrous formation by unfolding these proteins and having them form some sort of soluble aggregates so that they have better potential to form these networks when extruded under pressure and high temperature.
Right. Another one I have with the cold plasma treatments, you saw more soluble aggregate formation.
Yeah.
Right.
Yeah.
So do you think that helps to enhance the gel formation? Is it the gel strength, or how do you see the gel formation is improved?
Yes, so we confirmed the presence of soluble aggregates by running them on size exclusion under different conditions. I didn't show all the data. We ran them under like in buffer, in STS, and in BME, and then confirmed that in fact, yes, we saw higher soluble aggregates formation due to the treatment. And then we took these proteins and we formed a thermally induced gel. And we looked at the rupture strength by texture analyzer, and we saw a higher strength of the gel requiring higher force of rupture, basically.
Okay.
Yeah.
Good. Okay, good information and good investigations. Thanks.
Yeah, absolutely. Thank you, Janita.
I see a question from Lamia.
Yes, thanks, Pam, for the very informative presentation. Regarding the cold plasma, my question is, how do you see the application of this process? And is it after once we got the protein isolate, or where do you introduce this treatment to get more functional proteins down the road?
Ideally, just before spray drying of a protein ingredient. So basically, when we are extracting the protein and it is still in liquid form, and prior to pasteurization and spray drying, you introduce cold plasma. So treatment. So that is ideally where it should be, so that you don't spray dry and then put in solution and then treat and then spray dry again. So the idea is to do it as an intermediate step prior to spray drying. And the next step of the research is to actually looking at scalability. So my collaborator from mechanical engineering and I are planning to increase the scale to see if we can actually scale up to large quantities that would potentially be industrially feasible.
Okay, thank you.
You're welcome.
Yeah, Kostov, I see you raise your hand.
Hey Pam, wonderful presentation. Always nice to hear that. A quick question on the Mylar things that you were talking about, is changing and doing the glycosylation kind of things. Can you make a comment like what you see it's going forward, that production of acrylamide or heterocyclase amines, does it create an impact on downstream once we consume those kind of products?
Yeah, so basically the whole idea we call it Maillard-induced glycation because it's the first reaction of the Maillard reaction. So we control it in a way that it does not progress to form AGEs, advanced glycation end products. So the conditions are so strictly controlled so that it does not form advanced to intermediate and advanced stages, basically, of the reaction. That's a good question.
Thanks.
All right. I think we're at the end here.
Yeah, I think thank you so much for our two excellent speakers. Thank you to share your excellent research achievement as well as your inspiring insight into the new technology, new protein development, food safety and quality. Towards the end, there's a Sustainable Protein Forum in October 46, 2022. I encourage you to register for this conference. Also, as I mentioned that I encourage you to attend our annual meeting in May, I think 1 to 4, is specifically in PCP divisions. We have 9 sessions. We will talk about green processing, emerging source of the protein, biotechnology, protein functionality, bioactivities, as well as new food and non-food applications. I would like to see all of you there in our annual meeting. And also there's a lot of, networking opportunities. Maybe Beer, you can introduce these activities a little bit.
Yeah, of course. Yeah, obviously we'd love to see you guys there as we're all trying to come out of the pandemic. Hopefully we can see people in person for the first time in a while. But yeah, just if you can come in person, we'd love to see you. We have the PCP dinner is on Tuesday, May 3rd at the Hyatt Hotel. So we'd love to see you guys there. There's obviously a lot of other networking opportunities at the conference. There's a speed networking that's going to be really fun. We already have a lot of people registered for that. And there's lots of other networking, division networking events. So, and don't forget to, check out Inform Connect for, any sort of updates or to post. Like I said in the chat, if you have any questions that didn't get answered, go ahead and post them to the open forum in Inform Connect and get a conversation started and see sort of what kind of responses you get. So I just wanted to thank the PCP division leadership because they've done a great job with this mixer. I wanted to thank both speakers. They were extremely informed in a very short amount of time, which can be very difficult at times. So thank you to all involved.
Okay. And towards the end, we are planning for our scientific sessions for next year through the roundtable meetings. I would like to see all of you to get ideas and how we can organize our scientific sessions in a better way. I think Beer and other AOCS staff will send you email about the time and the schedule for our roundtable meetings. I hope to see all of you there. And also thank you for all the audience today. And we haven't, have in-person meeting, and at least this is online meeting for us to see each other face to face. Okay, it's the time to wrap up the meeting. Thank you, everyone. Have a good rest of the day.
Thank you
Thanks,
all.
everyone.
Thank you.
All right,
Bye-bye.
thank you. Bye-bye.
Bye-bye.